Enzymes are proteins that speed up chemical reactions by lowering the activation energy required to start a chemical reaction. Enzymes act as catalysts, which means they are not consumed in the process of the reaction. In an enzymatic reaction, substrate molecules bind to the active site on the enzyme. Next, the enzyme changes shape slightly to hold the molecules in place. This is called induced fate. Once the substrate is converted to the product, the finished molecule is released. Catalase is an enzyme found in large amounts in the peroxisome organelle of a cell. Catalase breaks down hydrogen peroxide into oxygen and water. Catalase in humans has an optimal temperature of about 37 degrees Celsius and an optimal pH of about 7. In this experiment, the question that we're going to try to answer is, does the amount of catalase affect the rate of reaction? Our hypothesis for this experiment is, if the amount of catalase increases, then the rate of reaction will also increase.
Materials required: 1 milliliter pipette, safety goggles, pastel and mortar, labeling tape, test tube rack and test tube, computer, Logger Pro, gas pressure sensor, electronic scale and weigh boat, distilled water, graduated cylinder, beef liver, hydrogen peroxide, cutting board, tweezers, and a razor.
Use electronic scale and weigh boat to measure 5 grams of beef liver. Label the test tubes 0 through 5 for number of drops of liver solution to be added. Add 5 milliliters distilled water and mix with the liver for 50% liver solution. Use a 1 milliliter pipette to add drops 50% liver to test tube. Using a new 1 milliliter pipette, prepare to add 1 milliliter of hydrogen peroxide to test tube. Quickly add 1 milliliter of hydrogen peroxide to test tube, cord test tube with gas pressure sensor, and collect data.
Data results. The overall goal of the lab was to measure how the amount of catalase affected the rate of reaction. Basically, the reaction of the varying amounts of catalase with hydrogen peroxide was measured using a Logger Pro pressure probe. The pressure probe measured the amount of oxygen produced over time by the anabolic reaction. To calculate the reaction rate for each amount of catalase, we chose 2 points in the section of the graph that had the most consistent gas pressure. Table 1 shows the times chosen. Table 2 shows the pressures in correlation with the times chosen for Table 1 for all the trials. Using the times and pressures from tables 1 and 2, slopes were calculated and are displayed in table 2. Then all of the averages were calculated. Using the amount of catalase and the average slopes, a graph was created to show the overall reaction rate of the enzyme catalase on the substrate hydrogen peroxide. Using the points on the graph, a line of best fit was created with an R-squared value of 0.7097.
Conclusion: our findings. Our hypothesis was supported because the graph of the average rates of the reactions showed that as the number of drops increased, the reaction rate increased as well. This increase in rate means that the enzyme catalase broke down the hydrogen peroxide into oxygen and water more quickly when there was a greater amount of enzyme present. This is because the greater amount of enzyme present, the greater the possibility would be of a substrate binding to the enzyme in its active site. This would occur because there would be more active sites available for the hydrogen peroxide to bind to. Weaknesses. Our first weakness is that the weight of each drop was not consistent since we used a 1 milliliter pipette. This affects the amount of catalase per drop and is shown by the graph because the rate of reaction was very inconsistent as the R-squared value was 0.7071 instead of 1 for a perfect consistency. Another weakness is that we had no consistent time frame for when we put the gas pressure probe on the test tube. We also did not grind up the liver the same way each time, and that would affect the amount of catalase per drop. We had uncertainties with the balances, and that would affect the amount of catalase that we had to grind up. Finally, we chose the starting and ending points to calculate the slope based on our own judgment. Therefore, these points could have been incorrect and the slopes would be also incorrect. Further experimenting. Firstly, we could use a micropipette instead of a 1 milliliter pipette to add drops of enzyme to the test tubes to ensure there would be the same amount of enzyme each time. Secondly, we could count 1 second before putting the gas pressure probe on the test tube for more consistency. Finally, we could choose points that were a little bit after the starting point and a little before the end point to make sure that it accorded with the most consistent rate.